Next, we will begin with the next part of this chapter, which is degradation, protein degradation. Now, under degradation, there are two steps. First one is transamination, then there is deamination, and then urea cycle. Let's first see why do we need to degrade the protein or the amino acid, and then how it is done. Amino acids can also be broken down to produce energy. Amino acid will undergo oxidative degradation or oxidative breakdown under which conditions? First condition is when the intake of protein is greater than the amount of proteins which are required by the body, which means that the diet is rich in proteins. Second condition is during starvation. During starvation or during diabetes mellitus, when the glucose is not available for production of energy, proteins or amino acids will be broken down for energy production or to meet the energy needs of the body. And third is simple, the body or the cells will directly utilize the amino acid for energy after the protein has been broken down or hydrolyzed into smaller amino acids. Now, hydrolysis of protein. First, protein needs to be broken down into smaller particles. Then these smaller particles will be again further broken down into still smaller constituents. First, let's see how proteins are broken down. Proteins which are present in the body or which are taken from diet will be broken down using enzymes. The proteins which will be taken via diet are broken down first in the stomach by the action of pepsin and renin, where the proteins are broken down into peptides. In the small intestine, an enzyme called enterokinase is used to activate trypsinogen into trypsin. Now this trypsin, chymotrypsin, carboxypeptidases and aminopeptidases, which are secreted by the pancreas, they will break down these peptides into amino acids. And all these enzymes have different products of breakdown. They will act on different amino acids which are present in the प्रोटीन. तो अगर ट्रिप्सिन है वो यहाँ पे कर सकता है, काइमोट्रिप्सिन बीच वाले मॉलिक्यूल अमीनो एसिड पे एक्ट कर सकता है, कार्बोक्सी सी एंड पे एक्ट करेगा, अमीनो पेप्टिडेजेस एन एंड पे एक्ट करेंगे प्रोटीन के. सो दे हैव डिफरेंट साइट्स ऑफ एक्शन ऑन द प्रोटीन. नाउ वी हैव अमीनो एसिड्स broken down. Let's see how this amino acid can be further broken down into smaller particles. The dietary protein say we have derived amino acid by the action of enzymes. Now if we see the structure of this amino acid, we can see or generalize the structure as amino group is present, and then we have a carb or carbon skeleton. So now we need to remove both of these. So this carbon skeleton is removed and amino group is also removed. The carbon skeleton will be converted into alpha keto acid, and this alpha keto acid enters the Krebs cycle, and we all know that Krebs cycle will ultimately be used, or the products of Krebs cycle will be used for production of ATP, or it can be used for production of different intermediates. Now this group of the amino acid can either be utilized for biosynthesis of amino acids or nucleotides, or If this is in excess, then it is converted into carbamoyl phosphate and excreted as urea via the urea cycle, which takes place in the liver. Okay. Now we'll see the fate of carbon skeleton and the fate of ammonia group. To break down the amino acid, two reactions take place. First is transamination reaction, in which the amine group will be removed, and the second is oxidative deamination, in which ammonium will be separated from the amino acid and released for the utilization for synthesis of different amino acids or nucleotides, or for excretion. The first step is transamination, second step is oxidative deamination. In transamination, as the name suggests, there will be transfer of the amine group. In this, alpha amino group is transferred from amino acid to alpha ketoglutarate. Alpha ketoglutarate, you can view this as a carrier for amine group. Okay, so there will be an amino acid and there will be alpha ketoglutarate. Now from this amino acid, this group has to be removed. This group is removed by the action of amino transferase, and a prosthetic group which is attached with it is pyridoxal phosphate, also called PLP. This is pyridoxal phosphate. Now with the help of amino transferase, this amino group will be transferred to the alpha carbon of alpha ketoglutarate. ठीक है, तो जब ये ट्रांसफ़र हो जाता है, उसके बाद कौन से प्रोडक्ट्स हमारे पास रहते हैं? जो अल्फा अमीनो एसिड है, दैट इज कन्वर्टेड इनटू एन अल्फा कीटो एसिड. ठीक है, कीटोन ग्रुप प्रेजेंट है इंस्टेड ऑफ द अमीनो ग्रुप, whereas the कीटो एसिड क्यों? क्योंकि एसिड ग्रुप भी है और कीटो ग्रुप भी है. इधर भी कीटो प्रेजेंट है एंड एसिड ग्रुप भी है. So this keto acid, which is alpha ketoglutarate, is converted into amino acid. ठीक है, तो यहाँ क्या हो रहा है? Amino acid 1 is combining with alpha keto acid, which is alpha ketoglutarate, to be converted into, this will be converted into alpha keto acid 1 plus amino acid 2. I hope you've understood this. Now let's talk about the different examples. यहाँ पे अमीनो एसिड लिखा है हमने और यहाँ पे कीटो एसिड. इधर अमीनो एसिड, सॉरी, यहाँ पे keto acid, and here we have the amino acid. So amino acid L-alanine is converted into keto acid 2, which is pyruvate, and this keto acid alpha ketoglutarate is converted into amino acid L-glutamate, and this occurs with the help of enzyme amino transferase, जिसमें prosthetic group है PLP, which is pyridoxal phosphate. Second example is of L-aspartate. Aspartate is also an amino acid. L-aspartate, when it combines with ketoglutarate, the amino group will be transferred to ketoglutarate, and this ketoglutarate will be converted into glutamate, which is an amino acid, और L-aspartate किसमें convert हो रहा है into this oxaloacetate. Again, enzyme will be amino transferase, and it has prosthetic group PLP, pyridoxal phosphate. This was the first step of the degradation or breakdown of amino acids, in which the amino group is transferred to some other molecule. The second step is oxidative deamination. In oxidative deamination, it will occur in the hepatocyte mitochondria. Glutamate, यहाँ पे example हमने लिया है, glutamate is converted into alpha ketoglutarate to release ammonium ion, which can be used for biosynthesis of amino acid, either nucleotides or different amines, or it is excreted via the urea cycle as urea. ठीक है, यहाँ पे जो deamination है, यहाँ क्या होगा, जो अमीन ग्रुप था वो completely remove कर देंगे, बट जो transamination था वहाँ पे अमीन ग्रुप is transferred from one amino acid to another amino acid. There was transfer, but in deamination there will be a complete removal of this amine group. Now let's see, यहाँ पे example है glutamate. Glutamate का जो amino group है, this amino group is being removed as NH4 positive or ammonium ion by the action of glutamate dehydrogenase. And when the ammonium ion is being released, it is replaced by a ketone group. Now this compound will be called alpha, because alpha carbon पे है, alpha ketoglutarate. ठीक है, ऐसे ही serine undergoes oxidative deamination and it gets converted into pyruvate, again by the action of enzyme serine dehydrogenase. This is an intermediate which is formed. डायरेक्ट आप इसको ऐसे भी दिखा सकते हो, by the action of glutamate dehydrogenase. Now this ammonium ion, if it is in excess, it can be transported by glutamine. ठीक है, अगर ये एक्स्ट्रा है, बॉडी में रिक्वायरमेंट नहीं है इसकी, either to synthesize amino acids or nucleotides or different amines, it will be excreted via the urea cycle. But to transport this ammonium via blood to reach liver, जहाँ पे इसकी conversion होगी via urea cycle, it has to be transported as an amine. An example we are taking of glutamine. So this glutamate which was present here, अगर यहाँ पे हमारे पास अमोनियम एक्स्ट्रा है, अमोनियम आयन की ज़रूरत नहीं है, this glutamate will be converted into glutamine. ठीक है, now this glutamine will be the transporter of the amino group, and it will be taken to liver via the blood vessels for urea cycle. Now let's see how urea cycle operates. Urea cycle will take place in the hepatocyte. So we had said that via blood the glutamine will be transported to the hepatocytes. When it reaches the hepatocyte, glutamine is transported into the mitochondria, and then glutamine is converted into glutamate. glutamate, and this glutamate will be broken down into NH4 ion and alpha keto glutarate. ठीक है, जो आपकी ये यूरिया cycle है, it begins from here. Now this ammonium group will be converted by the action of carbamoyl phosphate synthetase 1 into carbamoyl phosphate, and will use 2 molecules of ATP for energy and 1 molecule of H2O. Now this carbamoyl phosphate, इसमें से phosphate group निकाला जाएगा and it will be combined with ornithine by the action of citrulline transcarbamylase to be converted into citrulline. So ornithine and carbamoyl phosphate will combine together to form citrulline. Citrulline will be transported out of the mitochondria into the cytoplasm of the hepatocyte, where it will be converted into arginosuccinate using the molecule aspartate and ATP with the help of enzyme arginosuccinate synthetase. ठीक है? Now this arginosuccinate will be converted or will be broken down into fumarate and arginine by the action of arginosuccinase. Now this arginine will be broken down into ornithine and urea will be released. तो ये जो ये ammonium ion था, that will be present here in urea. ठीक है, so arginine is being broken down into urea and ornithine by the action of arginase. Now again, this ornithine can be utilized or recycled, so this ornithine is transported back into the mitochondria, where it will again combine with carbamoyl phosphate, which was derived or which was synthesized using ammonium ion, and then again citrulline will be transferred out. Citrulline will be converted into arginosuccinate using aspartate. Arginosuccinate will be broken down into arginine and fumarate. Arginine will be broken down into urea and ornithine. तो ऐसे यूरिया साइकिल ऑपरेट करती है, और अब ये जो यूरिया होगा जिसमें ये अमोनियम आयन प्रेजेंट है, it will be taken to the kidneys for excretion. So this is how the extra ammonium ion are being excreted out. Why is there a need for excretion? Because ammonium ion is poisonous for the body. तो अगर इसकी deposition होती जाएगी, the body starts to swell up and ultimately the person goes into coma. Brain भी swell कर जाता है, सारी organs swell कर जाती हैं and the person dies. So there is a need to excrete the extra ammonia. अगर extra नहीं है, this ammonia can be converted or can be used for synthesis of different amino acids or amines or nucleotides.